the pathology continue progressing, and affects thalamus. So, for example, a lot of people get pain after several years, get horrible, not really easy to manage pain in an arm or in another part of the body. And the most important thing is that these usually progress to the cortex, and it's when people start to have a cognitive decline and some problems with the memory. And this happens years after you start with Parkinson's disease.
Sounds like me.
So what ideally we want to achieve with this project and with this therapy in long term is decrease or stop this progression of the alpha-synuclein pathology. So in a way that we won't be able to cure in terms of rescue the cells that already died. They are dead and you cannot do anything unless you do an implant. But what we want is stop the progression. So then you can be like in more initial stages where drugs or deep brain stimulation can manage very well the motor symptoms and you don't have the other problems. And this is the ideal.
It's not that you can, if you can stop the progression, you can actually get an improvement because symptoms are not always associated with cell loss. They can be associated with dysfunction of the cells you already have there. So if you stop the progression of the disease, those cells that are left can improve their function and can actually improve the symptoms.
And mainly what happens is when you get all these symptoms, in like the cognitive decline and all these things, it's not really that your cells in that part of the brain are dying, it's that they are not working properly. So we think perhaps this can help to continue in a healthy state. So I will, Mark have been talk a bit about genes, but I will explain this as easy as I can. So what is gene therapy and why? So this is our protein of interest. So I will say this is the house. The gene will be the plants to build the house, and then we have one thing that we will try to target in our disease, that is the mRNA. And I will talk about like the builders. So what is the idea? So in some therapies that they are doing now, what you want is to increase the amount of a good protein. So what you do to increase the number of the plant, or you detail more. What we want to do when we want to decrease a protein, we will attack the builders. We will decrease the number of builders, and then everything will go slow. We will have less protein. Okay. So mainly this is a funny image. So what we want to try to do, going into the cell and modify the cell, cure what is not working properly. So probably some of you have listened about some clinical trials in Parkinson's disease nowadays, and there are 2 or 3 already in phase 2 with some positive results. And in most of these clinical trials, what they want to do is increase a protein that is protective, okay? Or a protein that is involved in the production of the chemical factors that decrease due to Parkinson. And they use these structured viral vectors. They are as the virus where you get an infection. And the main problems with this approach is that you need to inject the virus into the brain. The virus are not able to cross and go into the brain. And you only can treat a very small area of the brain. And there is a theoretical problem that could be if you need to re-inject, your body will recognize that there are virus and will respond like when you have a cold. First time you are healed, the second time you are healthy because your body read out the virus. So here can happen the same in theory. But this is why I say in theory, because nowadays nobody have done a re-administration in a clinical trial with adenovirus. So this is an idea that is there, but it's not clearly proved. But it could be one of the limitations. But main limitation is you need surgery, you only can treat a small area of the brain. So what is our optimal approach? You can inject like any intravenous drug. You inject here the drug, and the drug is able, your treatment is able to go directly into the brain. So it should be the gold standard for the gene therapy. And we have tried to get something to go there using exosomes. So what are exosomes? They are small vesicles like balls that the cells release to the outside, to the medium. And they were very interesting for us because in 2007, a group based in Holland proved that these exosomes by themselves are able to deliver the proteins and other molecules, like the ones we want to use for gene therapy, to other cells in a natural way. So what they say, they take exosomes for mouse and put in human cells, and they found new proteins that are, that are mouse, not human. So this is how they prove it. So we thought, oh, this could be a natural way to deliver our gene therapy molecules. But for that, maybe you need to modify the exosomes to target the brain cells. If not, they will go to other organs that we are not interested. And we have done that. So here, this is an image of an exosome. These red dots are our therapy, and these triangles is what we put in the external surface of the exosome to target only the brain. So we put something that the name is RBG, is a protein for the rabies virus, and you know that when you have rabies, you get mainly brain damage. So we use that strategy to go to the brain, but it's only a small sequence, it's not the virus, and it's not damaging at all. It's only a strategy to use it. So we put these small triangles that will be recognized in the cells in the brain. Okay, so they have like a key mechanism. So they will go there.
So is this an alternative to viral
yes.
delivery?
Yes.
Basically.
This is what we are doing. It's an alternative.
It's a kind of what is it? It's sort of pretending that it's something else.
Yeah.
You put a nice